Okay, now for another multiple alleles example that I think makes a little more sense than the chinchilla and the Himalayan, just by the way that we represent it. And that has to do with blood type. If you watch any CSI shows and they find blood, one way they can alleviate suspects is by looking at the blood type that was left behind. And so blood type has many different alleles. And so we know already that you can have blood type A, you can have blood type B, you can have blood type AB, and you can have blood type O. And so you have an allele for the A, you have an allele for the B, and then you have the recessive form in the O form. And so that's more than 2 alleles for a given trait, which makes it multiple alleles. So let's go through the possible genotypes for blood type. So if we look at someone who has blood type A, there's 2 possibilities. And we're going to use a capital I to represent that they have antigens, or little flags on the blood cell that identify whether the red blood cell is A or B. So in the first case, we have homozygous A. So we have the 2 big I's that represent that it has antigens. And then it's going to have a big A. So that's homozygous blood type A. The other possibility is that it has one allele that puts the flags on the red blood cells, and then it has another allele that doesn't code for the flags. And so that would be recessive, and that would be a little I. So we have homozygous, and then we have heterozygous for blood type A. The same thing for blood type B. A person with blood type B can be homozygous B, which means it has 2 alleles that are going to plant flags on all the red blood cells that say this is blood type B. Or it can be heterozygous, in which it has one allele that plants the flags and one that doesn't. It still has the flags on the red blood cell that says that it's B blood type, but one allele just doesn't code for those flags. So we have the homozygous B, and we have the heterozygous blood type B. Then we have blood type AB, and there's only one possibility for a person's genotype with blood type AB. They have to have one allele that plants the A flags and one allele that plants the B flags. So then you have a red blood cell that on the outside it has both A and B flags. And then the last possibility, which is more rare, is to have blood type O, in which you would have 2 recessive genes in which there's no flags on the red blood cells or no antigens that identify the red blood cell as being A, B, or AB. Okay? So our example that we're going to do in class is to cross a heterozygous blood type A with AB. Notice that I didn't have to tell you the father's blood type as genotype as being either heterozygous or homozygous, because there's only one possibility. So the first step is write the genotypes of your parents. And so I know I have one allele that's going to plant the flag. So I have the big I to represent it's going to plant the flags, and A to represent it's going to plant blood type A flags. And one allele that doesn't plant anything. That's the heterozygous version. Then I'm going to cross it with the father, which is going to have one allele that plants A flags and one allele that plants B flags, and that's going to identify that blood type as being blood type AB. Okay, step number 2 is to complete your Punnett square. Mom goes on the side. Okay, so now I'm going to write my possible genotype. So I'm going to start with my first square. So this is step 3. And I have I sub A, I sub A. That's simply saying I with a superscript of A. And I look and that's the only one I have, so I have one I sub A, I sub A. Then I go to my next one, and remember, it doesn't matter what order you write the genotypes. So when I rewrite this, I'm going to want to organize and put the dominant I sub A first. So I have I sub A, little I. And I look and there's no other combinations like that, so I have one of those. I go to my next one, and I have one out of the 4 possibilities of having the genotype of I sub A, I sub B. And then my last one, I have one possibility of having I sub B, little I. Okay, now for the phenotypes. A lot of people make the mistake in that when they're writing this out, they're going to put heterozygous and homozygous. Well, when you get your blood type done, you can't tell whether you're heterozygous or homozygous. I have to tell you that in the problem so you'll know what genotype to use. But when you're doing phenotypes, you're not going to know whether it's heterozygous or homozygous in the phenotype. So if we look at the first one, this first genotype is going to have the blood type A. Our second example is going to have the blood type A. The third one is going to have blood type AB. And the fourth one is going to have the blood type B. So this could be used for a paternity test. If this pair of people, the mother gave birth to a baby that had blood type O, then there'd be a 0 chance that it would be the father's child, because there's 0 probability based on his genetics that he could birth a child with this woman with blood type O. But we have to look at our phenotypes now because we have some that combine. So we have 2 that can be A, 1 AB, and 1 B. So there's a 50% chance of having a child with blood type A.